MOSAIC is an ambitious program to collect spatial omics data across thousands of patients in cancer. What spatial biology allows you to do is to basically pick out many different regions within the tumor and then dissect those at the molecular level. So the goal really is to go after 7 cancer indications and for each one of them collect 1,000 patients worth of information. This is a very large number of patients. These spatial biology technologies are brand new. They've only just started being used in many centers, and the largest studies that I've seen in the literature consist of a few tens of samples. So going to the level of 1,000 per indication is a dramatic increase in scale. The MOSAIC collaboration has several important goals. First, scientifically, we would like to break up complex cancer diagnoses into distinct subgroups with different biology, enabling precision medicine. Secondly, we want to use MOSAIC as an opportunity to really understand how to make the most use of spatial omic data of this kind, setting standards and trying out novel analytic algorithms to enable the dissection of biological information from spatial omic data. The way that we've decided to construct the collaboration is to involve many medical centers in collaborative research.
We have now a large portfolio of drugs. We must define which patient is most likely to benefit from which drug. And I will go even beyond. We need to define for each patient what should be the best theoretical drug. And for this, we need to characterize the biology in population of patients, but also in each patient, and this is what we call precision medicine. And what has been reported is that the way cancer cell and the immune cell are organized could define the sensitivity to very specific drug. And this is the rationale to develop spatial biology. Currently, we can only say the spatial distribution has some impact, but we can really not go to the clinical utility of defining the spatial distribution. So clearly, if we want to reach the vision of using spatial biology to predict outcome and to develop drugs, we need to go several thousand of patients, several thousand of samples, and several thousands of protein and gene.
Spatial omics is a technology that quantitates RNA or protein expression in a spatial context of tissues or cells. NanoString are pioneers in spatial biology. We have 2 platforms, GeoMx and CosMx, and GeoMx was one of the first automated commercial solutions in spatial omics field. I'm really excited to participate in this initiative because it's an opportunity for us to think really hard about doing spatial biology at scale. I think that spatial omics brings up new ideas and new opportunities and new questions that the community can develop upon.
What will be both exciting and challenging about the MOSAIC dataset is that it is multimodal and spatially resolved. What that means is that we're looking at an unprecedented level of complexity of information about every single patient. Data like that are extremely hard to analyze. And the methods that people have come up with to deal with complex data of that kind right now primarily rely on deep learning. Owkin is very well positioned to lead the MOSAIC effort. Firstly, because we have a lot of experience working with deep learning models of that kind. Secondly, because we've been looking at histogenomic information for many years, trying to understand how image features relate to molecular profiling features and how that relates to cancer biology.
So when I first heard about MOSAIC, my reaction was, this is exactly what we wanted to do, but we are not able to do. We strongly believe that the topic of spatial biology is so complex that the only way to move forward is through a consortium of multiple centers and scaling up the ambition of the project. So at Gustave Roussy, we are very happy to participate to MOSAIC. The aim of the project is to improve the outcome of the patient, and this is the mission of Gustave Roussy.
Now is an especially good time to engage in a project like MOSAIC. First of all, the spatial omics platforms are now at a stage where you can scalably generate a lot of data from a lot of patients. Secondly, immuno-oncology has gotten to the point where we can really try to understand how the immune system interacts properly with cancers and can be used to control the disease. We are excited to see other institutions join the collaboration and would love to work with academic and industrial partners to make this an ever more impactful effort.